Hello, how are you guys?
Hello.
Hello, how are you, Himanshu?
Yeah, I'm fine.
Hello, Susan.
Hi.
Hi. All right. So welcome to the meeting. So it looks like we want to do updates. I'll do Sushmanth and Himanshu in order, and then I don't know if Susan has any updates, but then we'll get to her.
My graph is still upside down.
Oh no.
No, yeah.
Can I start my update, brother?
Yeah, go ahead.
Yeah. Last week, I completely worked on the model architecture actually. I started the code of DevoNet model. Here, there are two folders here. One is library folder where whole code is there, model architecture, how we will take the input and all. Last week, I worked on the model and the loss function. Here, I took two models. For nucleus segmentation network, one model is there, nucleus detection network, one model is there. And yeah, it's completely written in PyTorch. I made some changes according to what I needed. And the loss function, I thought of using a direct line, but I thought a single line from PyTorch, but I thought it wouldn't be that appropriate. So I wrote my class for own dice loss function from net resources, and I'm working on that only. And last week I wrote last 4 week blocks also. Yeah. Can you see this thing, my block section? Is it shared or not?
It doesn't look like it.
Okay.
You gotta reshare maybe.
Here I explained every code what I wrote. Yeah. First I wrote the whole code here, and below that explanation of it is there. If someone comes up and sees our development code, some may not understand, new beginners. So I thought of writing a blog about it, every code I wrote. So whatever I'm writing code, I'm just explaining it in a clear way, in the points, what each word means in that. So that's what I am doing. I was trying to comment the whole code also, but right now this is my process, like GSoC week 4. I worked on the datasets file, 3D datasets file. I wrote every explanation of it, and that this loss function also I wrote about it. And this week I completely worked on the model architecture. So my main motive would be writing explainable code of model architecture of MSN and NDN. Yeah, I worked on these things only this week, and I tried to work on SAM also, but I couldn't handle both at the same time. This week I will try to manage model architecture with SAM model also. By next week, I'm just working on the model right now. There are no fine errors. I am using scikit library to image processing, because Mayuk once told me that he wants to use scikit image library for Devolum, but he couldn't use it of less time. So I started to use scikit image library for the Devolum. Actually, it is mentioned in Devolum issues page also that scikit image update to scikit image library. So whatever I was working on, I am writing in scikit image only. I am using a special method called watershed segmentation. It generally helps, like when two cells are overlapped to each other, it doesn't take it as a single cell, it takes as two different cells. So it would be more clear to us for finding the centroids of each cell rather than overlap. That's what my thing is going on. I'm updating it to Mayuk also every week. He's pretty closely watching the whole project.
All right. Yeah.
And Bradley, if you want to take a look on the blocks also, please take a look at it, because I tried to explain every word in a clear way that if someone reads it, they could understand it before. Any newcomers to Devolum or someone who wants to continue this project, they just understand.
Yeah. Could you put a link to the blog in the chat?
Okay. Just provide it. And yeah, that's what my update is. I completely worked on the model architecture. If you see here, you can see. You can see here. This part is for NSN model L2, and this
I'm not seeing your screen. You have to switch back to the.
Okay, sorry. Yeah, this part is for NSN. I use mainly convolutional 3D because whatever dataset we have is 3D dataset. So I use it 3D convolutional layers and try to implement it. And this part is of unit. I just want to compare it with our network and unit network. So I'm just trying to implement it. Mostly I will remove this part of code because my proposal was made upon NSN and Indian only. If I can compare these results of unit, NSN, Indian, I will try to implement it and see. First I was working on this. No, it's not similar to unit and 3D. I was just comparing the segmentation of all these three networks, SAM model and whatever I proposed in the proposal and unit model. Whichever gives the best results, that would be useful to us. I'm just checking on which segmentation network works fine.
So you're just making a comparison between the two types of models. So
yeah, just right. Okay. Proposing my model wouldn't be fine, right? Comparing with other models, and if my model performs well, I can just mention it like my model outperformed these models.
Could you describe the what a unit model is?
Unit model is mainly used for medical image processing. It has 9 convolutional layers in a U shape. Let me show you with an example. Yeah, this is how it looks. It's in the shape of, there are 9 convolutional layers. First convolution will happen, after that deconvolution will happen. And
you're not sharing your screen again.
Yeah, I'm not sharing my screen, sorry.
That's okay.
Sorry, yeah. These are 9 convolutional layers totally. First convolutional will happen until here, then deconvolutional will happen, and this is mainly used for medical image processing. See here you can see biomedical image segmentation.
Yeah.
And this is pretty old model. I'm just seeing which gives a better result, either SAM model or DevoNet model or unit. I will try to extract. I will try to compare it with the RCNN model which was implemented by Himanshu also. I will just compare the results and provide the graph which model outperforms which and which one performs well. I will take it as for the development. That's what my idea is. Just not giving out a model and telling it looks good, but with a perfect explanation which model outperforms which, and according to situation we can use the model.
Yeah, that's good. Yeah, that should be interesting to see which ones perform. You're going to use like a benchmark data set, or what kind of data set are you going to use for this?
I'm using cell tracking challenge data set.
Oh, okay.
And with that, I will just take a common. I will eliminate some images from the cell tracking, and I will just check the test results of all models on that. And after that, I will produce the loss function and IOU score, and I will just try to compare them. If with IOU score and the loss, we can just find it out which model performs well.
Okay. Yeah, that'll be interesting.
Okay.
Yeah, UNets are, yeah. I mean, they're probably older, relatively speaking, but they're also specialized for image processing. So
yeah, they are mainly for biomedical segmentation only. They are mostly used in cell segmentation, organ segmentation, etc. They are particularly for, microscopy images for that only.
Yeah, that's great. Thank you for that update. So this week you're going to be working on that comparison and getting the.
Yeah, I will. I was thinking to work on the training the model because the model is not completed yet. I tried to write the model architecture. So many errors I have encountered, but at last it is working fine. This week I will write the trainer code for it, and yeah, after that I will try to train the models. Actually, I was having GPU issues. All Kaggle GPUs are completed, which I have. So I bought some of GPUs from online from Google Colab of. Yeah, I'm trying to train that on that.
Well, that's good. Yeah, that's always a good resource, Collab, for doing that sort of stuff.
Actually, the resources are emptied because Collab provides up till like around I don't know, 50 GPU compute units, I think, so I don't remember. I completely used them off for this month, and I use it on Kaggle also. And the work has been stopped since 2 days, so I took another resources. I bought it 100 compute units. I will try to train the model. That's it.
Okay. Sounds good. Thank you. So hello, Dick and Jiahang and Jesse. Himanshu, are you ready for an update?
Yeah, I'll just share my screen.
Okay.
Is it visible?
Yes, I can see your.
Okay, so I was working on the model that I initially proposed about the mask CNN model, and I was able to figure out how to solve all the errors and basically create my own custom class for a segmentation dataset in which I could incorporate like the image mask as well as the bounding boxes for it. And after like a few error solving methods, I was able to finish this class of the dataset, which just returns the transformed image as well as the target. And the target has all the information which is needed for like segmentation. It has the boxes, the masks, and the area of the boxes, etc. So I was able to use this and successfully get the dataset viable for training. And then I basically created the model for like instant segmentation, as well as I was able to like use the bounding boxes, etc., and also create visualizations for it. So this is how it looks in the for the dataset that I've created. So, and I also trained it for like I used like the basic optimizers and schedulers, and I tried to train it for like 2 epochs, and like each epoch took like around 35 to 40 minutes because like the size of the dataset is a lot large. And then I just saved that model, and basically since I was just checking if I could train it and if the model fits perfectly. So I just trained it for 2 epochs just to see that, and I was able to like this is like the bounding boxes and for the predictions. So but like the actual masks, I've only got like only one mask, as you can see over here, because it was just trained for 2 epochs. So what my plan right now is to do like some hyperparameter tuning, as well as probably like make some changes in the data set. But like if I'm making some changes, I'll like talk with Sushmanth as well, because he's also figuring out how to deal with the big data set and all. And similar to him, I also have 0 compute units available, and which is why I was thinking of doing like some kind of parameter tuning on the CPU as well. But like after done making some small changes in the model, I'll just get some compute resources and just finish the training like on one night session. Like just keep my laptop running for like 10 hours, I guess. Yeah. And I've not used Kaggle until now, so I can just do the hyperparameter tuning on Kaggle, and then I'll revert back to Collab. And along with this, I started like working on the DevoGraph part of the project, like to find a better ways to derive graph embeddings. And right now, I'm just in the learning stage of it. So I also watched the the recording which was given of the video, like in which Jiahang like explained like what is the way of doing the preferential attachment, and also I'm doing that simultaneously as well. So the data set I've been working on is also the cell tracking challenge, and after after a like customized pipeline is created, I can work on the different data set for the membrane segmentation. And after that, I can just work on the data set which I had proposed like 2 weeks back, which was like for the larvae stage and which was like kind of elongated like this. So just to see how the model kind of, if it works on that or some changes have to be made, etc. So yeah, that's my update.
Well, that's good. Yeah, thank you. I heard you met with RV Raj. He's the student in working on another project in the orthogonal lab.
Yeah.
And how did that meeting go?
Yeah, the meeting was great, and he basically like kind of told me about the way he wanted the things to go, like to use Mesa instead of like the which we initially did for agent-based modeling, that is to use NetLogo, and I think that's the right direction to go, because like using Mesa is different from NetLogo because NetLogo has its own architecture and you have to follow some rules that NetLogo has for it. So creating the agent-based models for that environment itself is different than what we can do in Mesa. So like I gave him a basic walkthrough of what he should be doing and what should be the next steps, like to get some kind of a small algorithm, agent learning algorithm ready for like some for the reinforcement learning part of it. So like we could just do a basic while iteration and then move on to Q-learning just for a basic implementation of it, and figure out a way to just connect those both things, like the agent-based algorithm as well as the environment he'll create a Mesa. So that's where, and I also shared him some resources that I've used myself for RL, learning RL itself.
Yeah, thank you. We were discussing this Friday about kind of the plan forward, and he's also talking to Ankit Grover, who works in the lab. So Ankit is actually really interested in RL. So, that's going to be fun to kind of develop the RL capabilities there. So that's great. What are your plans for this coming week? What do you plan to do?
So right now I'll try to finish the nucleus segmentation part and then move on to DevoGraph as well. And in the meantime, I'll also look into what the open sustainability project is going. So okay. I'll try to attend the meet on Friday.
Oh yeah, that'd be great. Yeah, so Sushmanth has been at the Friday meeting. So we run a Friday meeting. It's at noon Eastern time in North America. So we do. We talk about the two projects in the orthogonal lab, but we also, can talk about the DevoWorm projects if necessary, and especially like if you can't make this meeting or for whatever reason. But we also have some crossover with the projects. So yeah, that'd be good. So thank you, Himanshu, for the update. So I don't know if we had Jiahang a couple weeks ago gave an update on his some things he was thinking about, and it was mentioned by Himanshu. So I don't know if Jiahang had a follow up to that or not. If you wanted to talk any more about that, or
yeah, well, hi.
Hi.
And you hear me?
Yeah.
Yeah, great. Long time to see, huh?
Yeah.
Yeah, hi. Yeah. Actually, I'm kind of busy this week because I'm busy as preparing for my degree confirmation reports, and it will be like after 1 month, and I have to present to some professors for my degree confirmation. So it could be a little bit challenging. So I spent some time on this. So maybe not much updates because I didn't have but thoughts on those on the research project that I mentioned 2 weeks ago, and I see someone mentioned that in this meeting today. So So firstly, like what I mentioned in 2 weeks ago, I said I wanna apply the topological data analysis to the analysis of spatial temporal topology within the cell development process. And it is a general general topic, and I haven't thought about any model design or some more specific details about this topic 2 weeks ago. And for now, I think there could be some approaches to solve this task. For example, the first of all is compared with other static system. For the cell development process, we have a topology which is dynamic. It is time dependent. That means we can explore the topology of like the cell dependence network or something else. For example, if we have a microscopy video and we have this video recording the cell development process, we can sample a lot of frames, and for each frame we can sample a lot of cells, like using some cell segmentation method, and then we have a points cloud, and we have some ways to, maybe we don't have to build a network, we can apply the positional homology, which is to explore the topological information underlying this points cloud. It could be approach, but if we leverage this approach, that means we have a static topology for each frame. But however, this topology is dynamic, so that means we need to find like some ways to build this dynamic topology. Like, because in my opinion, I think if we just consider the topology of each frame, and we do not consider the temporal connection between the topology across different frames, it seems like we are not considering the dynamics of this topology. So this will, if we do not consider these dynamics, that means we are regarding the whole stuff as just investigating the static topology of a network. That could be trivial. Like maybe I didn't make myself clear. Let me give you an example. For example, we have seen another paper in maybe 1 year ago, which is a topological graph neural network. And I'm not sure if you still remember that paper. And in that paper, the authors proposed a graph neural network to explore the topology within a static network. For example, you have a graph which is not dynamic, and you apply a topological graph neural network to explore the topology within this network. So I think in our case, the approach that I mentioned before is very similar to this topological graph neural network. So in my opinion, I think, so first of all, I can take some experiment to test how this topological graph neural network perform in our cases. And then I would like to try some methods to explore the dynamics of this topology. What I think this dynamic topology is very, very important, because you need to remember an example, which is the global rotation formed by the collective cell migrations that I mentioned in 2 weeks ago. And actually, if we want to see this kind of topology, which is a global rotation, or more specifically a circle, if you want to see such a topology, we actually need to look at the spatial topology as well as the temporal topology. Yes. So, and I think for most of multicellular systems, the dynamics is very important. So that means we need to find a way to investigate the dynamic topology. But first of all, I think I would try the topological graph neural network, that the simple approach is for the static topology. Yeah, they will be my first step. And for the next step, I would like to investigate the dynamic topology. Like there could be a lot of methods to consider the dynamic topology. The first approach is you can think of that for each frame, you have a static topology, and maybe you would use some kind of embedding or some vector to represent this static topology, and then we can just like other recurrent neural network or other works which focus on the time series data. Maybe we can use some recurrent neural network, or we can use some transformer to aggregate this topological information across different frames. I mean, this could be an approach. And another approach is, I think, a little bit more fancy, is because actually, I think persistent homology is a very cool, very great tool applied to the points cloud data. And I think if we have some ways to build such a points cloud across frames, like a single points cloud, covering several frames, maybe we can find some ways to use the persistent homology to investigate the spatial temporal topology. I'm not sure if I make myself clear. The challenge here is you have Euclidean distance, which is the spatial distance, and you can define some other types of distance. They are all spatial distance. But if we want to connect cells across different frames according to some kind of distance, that means we need to consider the temporal distance, like the distance across time dimension. It is not very intuitive like the spatial distance, like the Euclidean distance. So this could be one challenge is we need to consider how to define such a temporal distance. And the next challenge is how to unify the temporal distance and the spatial distance so we can build a point cloud, so we can apply the positional homology to such a point cloud. Yes. I think this just two of my, not sure if they are reasonable or they are meaningful. Maybe I would take a try after I finish my information. Yeah, I think, yeah, that's all. Yeah.
Oh, yeah, it's great. Yeah, thank you for that. Yeah, there we go. There we go. Thumbs up. Yeah, I mean, I talked about some of that 2 weeks ago. The persistent homology thing, I think, is really interesting from the standpoint of kind of taking these point clouds and breaking them down into a network, but also into some of these higher order shapes and things that might describe the data. So with segmentation, we have these point clouds, and we have point clouds in time, but the point clouds in time are interesting because they're connected. So like you have a point cloud at time 0, and then another point cloud at time 1, point cloud at time 2, and you have cells dividing. So you have sort of this regeneration of the point cloud as this more increasingly dense structure with things in different positions. And then the idea would be that you'd want to link those things. And we oftentimes we use lineage trees to do that, but in a point cloud, it's kind of hard to draw a lineage tree and then also say where are things moving in space. So we've done some of that with we've done some things with different techniques. We don't have a really good technique for it though. That's the thing. And so that might be a good way to kind of build a nice model of that change, but also the changes in position and all that. And then characterize it as like this change in spatial orientation or something like that.
Mm-hmm. Mm-hmm.
Yeah, so I mean, this is like kind of a lesson to people working on cell segmentation. That's where the data goes upstream in the pipeline. You go from like segmenting the cells to like analyzing sort of a first order analysis, which is the exploration of the point cloud. You can, label the point cloud points, as cells, and then you want to find the relations between the cells. And this is one of the things that we're trying. This is why we kind of come up with these techniques to kind of put together a model of the embryo, or at least the cells, and how they're sort of related to one another. So that's where this is going. And I like these kind of topological approaches because I think people have, people kind of know, at least in C. elegans, sort of what it looks like, in terms of where the cells are in space and then where they're differentiating. I mean, we kind of know how those things kind of unfold from the data, but we don't really have a higher level description of like what kinds of structures are maybe precursors to tissues, or what kinds of migration oriented things are emerging in the embryo. Those are kinds of things we want to maybe describe with these topological techniques. That being said, I'm not an expert in topological data analysis, and my impression is that there are these methods that exist, but they're not like they're probably new techniques that can be invented. And so yeah.
Mm-hmm.
But I think it's based on like homology and some of these other geometric approaches.
Yeah. Actually, for this, my motivation. But first of all, my motivation is actually, I think, topological data analysis have been widely applied to the biological stuff. But I think one interesting thing is, I think deep learning methods, deep learning approaches like the convolutional neural network, graph neural network, they have also been applied to the biological analysis as well. But like there is a gap between the TDA and deep learning approaches, especially the graph neural network. I think graph neural network. One interesting point about graph neural network is this approach consider the data as a set or as a points cloud, and that means from this kind of perspective, you can consider the geometry properties and the topological properties of the data. Yes, so it could be different from the like convolutional neural network, which consider data from the perspective of visual information from the images. So this could be different. So I'm the motivation for me for this project is I want to see if there can be some like interesting stuff happen when we like integrate graph neural network with the topological data analysis. Yes, on the biological stuff. Maybe I can make myself more clear. Like we all know that's the multicellular system. For example, the cell development process, they have rich topological information, and we know that we have a lot of approaches, such as the TDA, which are very good at exploring the topological information from data, like from the microscopic video. And we have graph neural network, which are very useful for like capture information from the big data, from a lot of videos when the data is very large, data size is very large. So, I mean, they have their own like advantages for the downstream task. But actually, I'm a graph neural network guy, and I'm more familiar with graph neural network. And I see that in the field of graph neural network, actually, I would not say the graph neural network have been widely applied to the cell development analysis. I would not say that because I didn't see too many examples. So first of all, I would like to see how graph neural network can be applied to the cell development analysis. And secondly, I would like to see when we consider the topological information in the cell development process, how the graph network would perform, like how graph network would perform with the help, with the support of topological data analysis. And things like this. I'm looking at some intersection between these two fields, like and how you would perform on this multicellular system. And from another perspective, I think is I think what I want to say is I want to like explore any kind of explanation of the deep learning technique from the geometric and the topological perspective. Yes, like for now there are lots of scholars working on the geometry or the topological techniques within the context of deep learning, and they found that these kind of approaches are very helpful for us to explain why deep learning could work and why those neural network could work. So I'm also very interested in this. So that is another reason that why I want to look at the intersection between the graph network and topological data analysis.
Yeah. I was thinking actually as you were talking about like a lot of the things that people do in 3D modeling, and that's highly, topological and geometrical. One of the things that you do is you create an object and then you extrude it in space. So you pull it up, you pull it out and you give it some depth. You have to define the depth. And what tends to happen is that you can, sort of manipulate the shape according to what you want. And then, so I'm thinking about like some of the things that happen in development and, having these sort of extrusion processes. And they're not like, they don't call them extrusion processes in the literature. There's like transformations like evagination or gastrulation or things where there's a change in shape in the embryo. And so, it's like they don't describe it. They describe it as like a process and they observe it. But there's like a computational element there that could be like interesting. And not just extrusion, but like a lot of these like simple complexes that you would sort of discover in a network. How do those change as we change that network point cloud, as we get cells dividing and they're changing their orientation. And so you basically, if you have like simple complexes are just like shapes that form in the network. So it could be a triangle or a diamond. And then that diamond would sort of track with the cells dividing and that network expanding so that you might end up with like maybe a hexagon that shifts its orientation maybe 45 degrees. And then it maybe curves a little bit on the bottom. And keep following that shape out as it's moving around, extruding, and being warped in different ways. So that's the kind of thing too that we could think about, like, what is we start with maybe like a very simple shape and a very maybe an 8-cell embryo, maybe like a diamond of 4 cells that kind of are in that part of the embryo, or maybe like the anterior end of the embryo. And then as the number of cells increases, as you get daughter cells and they change their position, they migrate, they sort of form these structures that start to, tissues, those will change the shape. And you can actually track that shape as like an object that's, it has properties just like a 3-dimensional object in something like Blender or Unity or something like that.
Yeah.
Yeah.
Mm.
Yeah, I mean, it's just, yeah.
I'm thinking about this. Yeah.
Yeah. Anyway, yeah. So that's great. Yeah, thank you, Jiahang. I think there are a lot of things that, like, following up from our discussion 2 weeks ago, and I wanted to get the people doing. I want to get people like Himanshu and Sushmanth thinking about, especially Himanshu, about like where are we going with this stuff? So we're going somewhere that's very kind of conceptually hard. And so we want to have, like,
Yeah.
I think, a good sense of where that is and what's possible. So yeah.
Actually, I think, for example, just because actually for this project I'm working on a graph network, topological data analysis, and I'm less good at the segmentation or related stuff. So first of all, for these research projects, if like anyone interested in this, and I'm very welcome for anybody reach out to me. Another point is during this whole GSoC period, if you have any related questions to the topological data analysis and the graph network, I'm also welcome for everyone, like, reach out to me. Yeah.
That's great.
Yeah. Mm-hmm.
Well, thank you, Jiahang. Okay, now I want to explain something that is related to the conversation that Jiahang and I had about networks and topological data analysis and GNNs. So one thing I mentioned was the simple complex that you find often in networks. So let's consider a simple network here where we have connectivity across 5 nodes. Okay, so this is a network topology where you have this triangle and these 2 lines that come out. Now, in a normal network analysis, we might be interested in calculating like the centrality or the clustering coefficient or things like that. But in a topological network analysis, you might be interested in finding shapes in the network. So what kinds of things are enclosed in the network topology? So in this case, we have a triangle, which we can define here. And if we add in nodes like this, we might have a couple more nodes that connect. We might have another triangle, actually 2 triangles. And we also have a rectangle, which is here. And so these are what they call simplical complexes. And these are fine, people are able to discover these, and these are kind of ways of flipping the typical network analysis on its head and saying, instead of maybe finding communities or finding centrality or finding a connectivity regime, I may be interested in finding shapes that are formed by the network. So this is important in sort of the spatial networks that we're dealing with in the embryo, this point cloud that we talked about earlier, which is in 3-dimensional space, or actually 4-dimensional space. We have this point cloud of points that are these centroids. And these centroids are kind of worked out in this way so that you have, they're always changing. We talked about this where this point cloud is always changing. We're getting new cell centroids, we're removing old cell centroids. And so this topology is always actively evolving. And so this point cloud actually actively evolves in terms of its outline, in terms of its structure, and things like that. Okay, so this is the point cloud. So this point cloud is constantly evolving. And as we said in the meeting, in the main meeting, we have these 2 views. We have the static view and the dynamic view. And so if we look at the point cloud or anything from those 2 points of view, well, let's think about the network. We have a static network, which is, again, something like this, where we have a triangle. This is our triangle. And say that triangle changes over time. So maybe this node kind of comes out here and gives birth to 2 daughter nodes. So we have something like this. So this node here was here. It's now removed. And you have 2 daughter nodes in its place. That means the connectivity changes. So that instead of a triangle, you end up with this sort of rectangle with 2 nodes here. And maybe this node actually dies, and you end up with daughter cells here, where you end up with maybe not a triangle, but like a fan motif. So you have this fan, and then this might be connected later into two more triangles. But let's focus on this image, this part for now. You have this triangle, and it transforms into a rectangle. So that transformation then is dynamic. So we can take two static views of the network on two different points in time, and end up with this dynamic shift from a triangle to a rectangle. Or a rhomboid, I guess, would be a better way to describe it. And so these two shapes, this transformation of shape, is actually what we're dealing with in development, because we have our embryo and it's changing its shape constantly as the cells are dividing. So we've moved from the point cloud to these kind of structures. Now, one thing I was talking about was, like, say we have like like a diamond as a cyclical structure. You know, we have this four tuple cyclical structure. I guess you define it as you have 4 nodes here, and this diamond might not change shape to something else. It might actually transform its sort of form. So, like, this thing here is flat. This is in 2 dimensions. But let's imagine that the cells change their position. You get daughter cells that emerge around it. So there's migration of cells. There's a change in the constituents of the network. You have a diamond that's warped into a 3-dimensional shape that now has sort of, it's extruded out in this plane, and then also in this plane. So it's kind of hard to see. I've also made a mistake in my drawing, but basically you have this shape that's changing its form. And so this is where I was kind of going with this. This is in 2D, and this is a static view. All right, and then this is where you're going into 3 dimensions where there's extrusion. And that extrusion maps to some developmental transformation, like maybe something like evagination or some process like gastrulation. And so we can actually take these geometric transformations, map them to the developmental transformations. And this actually goes back to Darcy Thompson's work way back in 1917. And, you know, sort of the idea was that you had, like, you could take a fish and you could put it on a grid where you could, each landmark would have like a certain point on the grid. And that tie point then would allow you to transform the grid or transform the phenotype, and you'd have a corresponding mathematical structure, or you'd have a corresponding morphological structure. So it was like a morphological structure, like a fish. So I'll just draw this out. You have a fish like this, and this is a very simple drawing of like some sort of fish. The point here is to show like what this phenotypic transformation looks like. So we have rays, which are kind of overlapping here. And you have this phenotype, and you have this grid behind it. So this phenotype is embedded in a grid. This grid has tie points, which are basically where the landmarks are. So this grid goes underneath the morphology. And there's a lot, you know, this is a very simple representation. And so you might have tie pointers, the eye, you might have tie points like this underlying the fish. And so each of these tie points corresponds to a certain landmark on the fish's morphology. As this morphology changes, if you look at, say, like, for example, another species, these changes move around this grid, so the grid gets warped. So the grid actually changes its form. So it goes from something straight like this to something like this. And so you can see what the topological transformation looks like. it's very much from a linear to a nonlinear sort of way. And this is, of course, changes in species, but it could be changes in time. It could be a developmental phenotype like an egg or some sort of larval stage organism. So we can do this sort of warping with developmental forms, and then we can look at this relationship between embryo networks or some sort of network topology and these shapes and how they change their form over time. So yeah, do we have any comments about that or questions? Don't know if how Dick is doing. How are you doing, Dick?
I'm doing better today.
Good. That's good.
Okay.
Yeah.
If you want, I'll present that strange new image processing problem.
Okay. Yeah, that would be good. Actually.
Okay. Okie doke. I came across it because there are now some X-ray sources which will produce a narrow beam of light or of X-rays. Okay, and I realized that in trying to solve the problem, there's a much simpler problem which apparently has not been addressed in image processing. So let me share my screen. Oops, share my screen. Okay, entire screen, and okay, go here, share. Okay,
There we go.
visible now?
Yes.
Yeah. Okay. Okay. Now I'll switch to AutoDraw to show you what the problem is. Start drawing. Okay. Take any picture whatsoever, which I'll represent as a square, and let's draw a line across it. Okay. Suppose that we sample all the pixels along the line. Okay. Then draw another line and do the same thing. We sample all the pixels along the line. And then another one. And another one. Okay. Now what this does is it divides the pictures into regions, and we could start to do what's called in-painting or infilling. Okay, interpolating between the values on the lines. This will then produce a picture of some quality. And if we have enough lines, I mean, it's clear. Here, let me do another one here. It's clear from the old-fashioned CRT televisions that if you have enough lines, you can get a good representation of a picture. And that's what television was based on originally until the flat screens have showed up. Okay, and so those are all parallel lines. But I'm saying let's take a random set of lines, use an in-painting algorithm, and that produces a picture. And the question then is, what is the quality of the image versus the number of lines? Okay, now the lines can be random, or we can do it more cleverly and say, we're going to add a line, but it depends on the quality of the image and where we put it. So you could then have image-dependent addition of lines, which would then do it with in-painting, and then give you a further improvement in the picture. So the question is, with random lines or with image-dependent lines, what is the quality of the image versus the number of lines? Okay, that's the general question. Okay, and it's a bit strange because you're taking these lines and you have a function along each line, which is the value of the pixels, and then use the infilling algorithm to fill in the picture. Now, there was an old problem of compressing images for transmission, so that they went much faster. It's not a problem nowadays, but this would also be a possible solution to image compression. And I can't find anything in the literature except the old-fashioned TV screens, which do anything like this. So it seems to be a new branch of image processing. Okay, so I'm looking for someone to work with me on testing this idea out and seeing where it goes. Okay?
Yeah. Yeah. So
It's
this.
a little abstract, but it came out of the X-ray sources, which generate lines across objects. These would be X-ray lines instead of visible lines. But backing up, it's obvious that the general problem, this combination of lines plus in-painting, is a form of representing an image with a lot less data than the whole image. Okay, so that's the problem.
That's great. Yeah. We talked about that with respect to the breast cancer work and how.
Yeah, the cancer work inspired it, but I'm sort of backing up and saying there's a general problem here. And if we can solve the general problem, it'll help the X-ray problem also.
Yeah. Okay, yeah, so this is a more general sampling problem almost.
Yeah, it's more general because you get all the pixels along the line. Yeah. With X-rays, you only get the sum of them. Okay?
Yeah.
I'm seeing if I can jog Jesse here.
Oh, yeah.
Anyway, I'm looking for a partner in this who wants to. It's much simpler image processing than the segmentation problems.
Yeah.
Okay, but it seems to be a brand new topic for image processing.
Yeah.
And who knows where it'll go.
One area I could imagine immediately is imagine if you had an image with a lot of noise in it and you couldn't unmask the noise. Yeah, so you had things like a bunch of noise as a mask over an image and you couldn't tell what the signal and noise, like you couldn't decouple them. So, you'd have to do that. You could do this kind of sampling where you go over, basically you'd end up sampling things at random. You just kind of pick a pixel and you flip it over and say, what's the information content of this pixel or that pixel? If you did it this way, you could like kind of divide up the image. So you could send a signal across, you could send a signal across here. Then you could find the intersection after you have enough lines and like explore that space.
Yeah,
it's almost like
an example of an application of this would be if you use multiple satellites and send signals between them, and the signals intersect the atmosphere, you can reconstruct the atmosphere from a set of lines between satellites.
Yeah.
So that's an example of a practical application. Okay. Another one is geological. If you have a shaft and you slowly drop a transmitter of some sort, and then you have another shaft at some distance from it, and you raise and lower a receiver, then you get lines between the two shafts, and you can reconstruct the Earth between the two shafts from a set of lines.
Yeah.
Okay, that's another example. Okay, so there might be there are a couple of practical implications, not just the old problem of image compression.
Right. It's almost like a divide and conquer algorithm on a space like that.
Yeah, I mean, image compression we solve by just increasing the bandwidth.
Yeah.
Okay, and then you don't bother with it. Okay, but it's still a curious problem, reconstructing an image from lines across.
Are you there? People. Okay. Oh, yeah, you dropped off there a little bit. Did you? What is the last thing you said? You dropped off.
Oh, said Jesse, send me a note, or I'll send you a note.
Oh, okay.
Okay, we can discuss it if you want. You might be interested.
Yeah.
I mean, it's not often you get to start a new field of image processing.
Yeah.
I think I've done it once before in my life.
Yeah. Yeah.
Okay.
All right. Well, thank you. That's great.
Okay, sure.
All right. Anyone else wanna have any updates, or Susan or Jesse? Any comments?
My imaging problem is a bit of a strange one. I have X, Y, and Z axis, and I have let's see, this is the Z axis, and this is the X axis, and then I have the Y axis going this way.
Yeah.
You know, you can see that.
Yeah.
And then so my diagrams are 90 degrees flipped from the paper I'm trying to follow, and I suspect that their y-axis is going out of the page instead of into the page, and so things are 90 degree flipped. Does that make any sense to everybody here? Would that flip a graph? I don't know.
Would it flip a graph, or I don't know how?
Yeah. Anyways.
I don't know. What program are you using?
Console.
Okay, so it's just plotting it in a way it's a little bit different than like usually the Z is the depth, but this is, you're saying
Yeah,
Y is
but if you have this where you have this is your X, this is the Z, and this is the Y, you can flip it so that's the Z is coming out of the page, the Y is up, and the X axis is going that way. So
Okay.
it's a more conventional system,
Right.
only it's rotated.
Oh. Yeah. I don't know. I've not run into that. I guess Himanshu said it would flip a graph, I think, but both, or let's see. Yeah. It would flip a graph, I think, but both Y and Z would be affected, right?
Yeah.
I guess, yeah.
Maybe could I show you the problem?
Yeah, if you want to, yeah, if you can.
Okay, is that better?
You can see, yeah, you can see your desktop now, so you should be
Can
able
you
to
see my?
There we go. Yes.
Okay.
Yeah.
Now, so this graph that looks like a mountain here, a pink mountain. I was trying to duplicate this, and I get graphs that are like this on the left-hand side, and I took this blue line here, this it's at 0.4 strain, and I moved it around the axis. I just changed the negative and positive. And I don't get, it's got a curve going up. It doesn't have this curve going down like this. In other words, it's kind of 90 degrees flipped. It needs to be turned over.
Right.
And then I looked at this, and this is rubber, and it looks kind of like my curve in the first quadrant. Anyways, here, I'll try to find the other one. Oh, and then I have this, and this is for a tall prism instead of a short one. Anyway, I got this to work for me here because I had my numbering system backwards.
Oh.
I had, yeah.
Yeah.
It's interesting. I got it for it. Numbering system is backwards here. Here, I don't know if you can see it in the corners, the X, Y, and Z, and the Y is going into the plane.
Oh, okay. Yeah, I see.
And if you somehow number it so that the y is going out of the plane, then you seem to get a flipped curve.
Oh, so the
You
number
Yes.
it's basically flipped from positive to negative, or it's negative going versus positive going.
Yeah.
Yeah.
Anyway, that seems to be holding me up, and
Couldn't you
I've
just?
spent many weeks working on this, and I keep getting this type of thing. Yeah, it needs 90 degrees flipping. Anyway, it's a really stupid problem, but it's consistent. I get it. This one for a really tall triangle prism. This is sort of a medium tall one, and this is for a shorter one. You can see there's a theme here. All kind of not, it doesn't follow this hill. It's not a hill, it's more of a swirl.
Right.
Anyway, that's my problem. So I just, it's stupid, and I'm hung up because I'm supposed to duplicate this paper or explain why it doesn't work.
Oh.
So any thoughts on this would be really great.
Yeah, I think that's some interesting data, and it's always hard to like get to get the graphs to plot correctly because they use their own conventions. Like, each program uses kind of a way that it puts in data. So, like, your typical like MATLAB approach is to use like a vector or some structure, some struct that you can plug in the values to the axes. For the XYZ though, couldn't you just transform the y-axis to something like?
Yeah, I was thinking of just turning it. There's a method of doing it, so I was just going to change that around if I can figure out the code. Like there's hidden, there's a whole pile of hidden attributes in COMSOL.
Oh, right,
It tries to do everything, and from my perspective, it ends up doing nothing.
Well, I was thinking just a simpler case of like 1 over x, and that would give you like a decimal, but it would be like transformed y. So it would like turn it around, but it would be a weird number, like a weird interval, but it would be transformed y. So it basically give you that orientation, and you just this is transformed y, and it's going in a different direction. So I don't know if that would work, but something like that.
I think that COMSOL does have it where the y direction can be changed.
Okay.
I looked it up, and there are, you can even put it in a spherical coordinate system. And if you get desperate, maybe I should just do that.
Maybe, it's just kind of now, you can
that, is more general.
Right. Now, when you rotate them, and I don't know if you're going to be rotating them in the presentation or what, but that's another thing to consider.
That's why I've been having problems.
Yeah.
That's exactly why I've been having problems.
Well,
so I'll stop sharing.
Okay.
There.
Well, good luck with that problem. I don't know. I don't know what else to say about it.
Yeah.
Those are some nice graphs, though. That was a good collection.
Yeah, at least they're consistent.
Yeah.
And going in the wrong direction. Yeah.
So, all right. I had a few things to share here myself. So let me share my screen. We're at the top of the hour, but it shouldn't take that long to talk about. So one thing I found this week was this interesting, this is having to do with using genomic data as sort of like an input for what they call foundation models. So this is, I think, from Stanford, a research group at Stanford. And what they're doing is they're working with HyenaDNA. What they're trying to do is build what they call long-range genomic foundation model. So this is a model where you can train a model on DNA, like genomic data, a bunch of nucleotides, and it can predict or make predictions for different types of DNA stretches. So one of the problems that we've had in genomics is DNA alignments, gene alignments, and then discovering stretches of DNA. So in the past we've used things like BLAST. We've used all sorts of mapping software where we're mapping between a reference genome and a genome that we're sequencing, and to get those aligned and to get them to know where they are in the reference. So this is a little post on this. We're excited to introduce HyenaDNA, a long-range genomic foundation model with context lengths up to 1 million tokens at single nucleotide resolution. So this is just a term that's, tokens are like what they use in this model to, provide this context length. HyenaDNA is pre-trained on the human reference genome and sets new SOT-A on 23 downstream tasks. This is, I think, state-of-the-art, including predicting regulatory elements, chromatin profiles, and species classification. So they can take they have a reference genome that they train the model on, and then it's kind of like a large language model where you're predicting on a bunch of letters. You're finding features like regulatory sequences, chromatin profiles, if you want to find a chromatin, different sites for chromatin activity, and species classification, so you can detect like a certain stretch of DNA. And usually we can do this in BLAST, where we find potential matches for some piece of DNA that we put into a BLAST search. So this is, this will give you those predictions. I don't know how it compares to BLAST, they don't really say, but basically that's what they're trying to do. It's like kind of a next generation BLAST, and kind of using a large language model large language model techniques to sort of get at this problem. We also explore what new capabilities open up with long context in genomics, including in-context learning with soft prompt, tunable tokens, and instruction fine-tuning. So this is the archive paper here. This is HyenaDNA. They talk about the paper. This is what they call it, a foundation model. Let's see. They do actually have some interesting things with respect to other types of machine learning models. So leveraging Hyena's new long-range capabilities, we present HyenaDNA. They talk about hyenas in here, which is an approach that they developed. Hyena, a large language model based on implicit convolutions, was shown to match attention, which is a method that they use in machine learning and deep learning, in quality while allowing longer context lengths and lower time complexity. So it's a large language model that they're using. They're fine-tuning it with respect to the data here. Leveraging Hyena's new long-range capabilities, we present HyenaDNA. So this is a subset of hyenas, which is the model that they have here. A genomic foundation model pre-trained on the human reference genome with context lengths up to 1 million tokens at the single nucleotide level. So they train it at single nucleotides, and up to a 500-fold increase over previous dense attention-based models. So people were using attention-based models for some of these genomic problems earlier so people have been applying machine learning and deep learning to this to sort of get improvements over traditional BLAST searches, and they're actually improving over these attention-based models. HyenaDNA scales subquadratically in sequence length, training up to 160 times faster than Transformer, uses single nucleotide tokens, and has full global context at each layer. We explore what longer context enables. So what that means is it includes first use of in-context learning in genomics for simple adaptation and novel tasks without updating pre-trained model weights. So basically, they can find very specific sequences. They can find novel sequences, and it doesn't need a lot of updating in terms of training. So this is a pretty flexible model. On fine-tuned benchmarks from the nucleotide transformer, HyenaDNA reaches state-of-the-art, which is SOT A, on 12 of the 17 data sets used, using a model with orders of magnitude less parameters and pre-training data. So they're able to train it on, their reference genome. They're able to get a good result on 12 of 17 data sets that they tested against, and they're able to use fewer parameters and pre-training data in general. So this is an improvement on some of these methods. So this is really about, getting this going from like blast searches, which were kind of so, 2000s to, different machine learning techniques to now this type of model. So they were talking about long context models, which are a state-of-the-art thing. It's very trendy. OpenAI stated a goal to reach 1 million tokens. These are these long context models, while Magic announced they've reached 5 million tokens for code. We're all for it, but most are focused on natural language and code. So this is where a lot of large language models focus on like using English and predicting text in English. They're using something in biology. They're using DNA sequences. And so we have, of course, 4 nucleotides that get rearranged in different ways, and you have these long stretches of DNA. And there are actually quite a few possible combinations that it can be. So it's a hard problem to solve. And you have things within those stretches of DNA. So you have different patterns. You have repeats. You have of different lengths. You have different types of combinations of codons and things like that. You have stop codons. You have start codons. And so you have a lot of things in the structure of DNA sequences which need to be predicted. And so it is people have made the analogy between DNA and language before, but this is where you're actually using this to find to improve the search process. So to give a sense of the scale, the human genome is 3.2 billion nucleotides and can be seen as characters in the sequence. So they have some blog posts on this as well. So they train this hyena model, which is a convolutional large language model. And so they've actually been able to do this with more generally, but they've applied it now to genomics, where they want to find, they want to really kind of put this to the test. So here's the sample so they have a reference genome. Usually you do this with some sort of reference genome that you build. So the first human genome, first human draft of the human genome is kind of like a reference genome. And they do this for different species where they kind of sequence a bunch of organisms and they find a consensus for the sequence. And then they put a draft out. And then that's the sort of the reference genome. And then if you go to sample a genome from your own sample, that's your sample. And then you try to map it to the reference genome so you can actually align things and make sure that they're in the right place in the genome. So that's what they're trying to do. But they're trying to pre-train the model with a reference genome and then throw data at it from different samples and see if they can make the predictions. So this is how it works. Basically, they have this reference genome. They use that as the pre-training step. Then they have these predictions, these token predictions that they propose. They match it up with this technique here. And then they have these downstream tasks where they try to classify things, structures within the DNA sequences, like finding regulatory elements, which are these, sometimes they're repeats, sometimes they're very specific sequences, domains within the DNA. Then they have chromatin profiles. So, you might have certain 2 or 3 DNA sequence or nucleotide sequence motif within your DNA sequence that you can discover. And these are potential sites for chromatin binding. Then you have in-context learning, which are, other features that you want to learn about. And that's really, sort of the heart of genomics. That's why we do genomics, is because we want to find these patterns that we want to say, like, this is useful for, some biological function, or, it's maybe a new gene or something like that. So, this is they're really kind of moving this area forward. And so they're trying to learn all sorts of things. They want to learn maybe to define like coding regions from non-coding regions. They want to find out more about gene expression. They want to find out about other types of structures in the DNA. And they ultimately want to go to building, going from DNA to proteins. So, this is where you have a sequence of nucleotides that gets transcribed into RNA, which has, usually there's some changes in the RNA that derive from that transcriptional process. Then that RNA gets built into proteins through another process of translation. And so, this is the first step in predicting what these proteins will look like. If you can predict patterns in the DNA, you can then take samples of RNA, predict patterns, and you can use something like RNA-seq or something like that, predict patterns in the RNA, and then link those two things, and then link that to the proteins, because now, we can, of course, model and sequence proteins. We can actually make predictions on that. And I think people are applying large language models to proteins as well. I'm not familiar with how that's, what the state of that field is, but so, you have these different DNA motifs, promoters, silencers, enhancers, and you want to be able to discover those in the sequence in context. So I'll provide this paper or this in the Slack, and if I can send it out in an email as well if people want to read it. Let's see. So let me check the chat here. Okay, so Jiahang had to leave. And if you're interested in what Jiahang had to say about networks and topology, don't hesitate to reach out. He put his email in the chat. If you're interested in TDA and GNNs. And yeah, Susan will keep us updated on the graphs. So I think that's it for today. Any other comments or questions?
I'm good.
Okay. All right. Well, thanks everyone for your discussions today. It was very good. Very good meeting. Yeah, thank you. All right, everyone. Have a good week. Thank you.
Bye.
Bye. All right, now I'd like to talk about sort of a third installment in the discussion we've been having about different types of embryoids. So 2 weeks ago, we talked about human embryoids and some of the work that's been happening there. And then last week, we talked about monkey embryoids in non-human primates. So we have monkeys and humans. And so we've been able to demonstrate this in 2 model organisms in primates. This week, I'm going to talk more about human embryoids and some of the gastroloid work that's been done. So, this is like I showed a diagram 2 weeks ago showing sort of this progression from organoids to gastroloids to embryoids, and just kind of how those models are, they're kind of refining those models to give us answers to different things. We could look at different things in development and get certain answers depending on the type of model we build. There's a lot of control here. It's either transgene control, transgenic control, which means you put a gene into the cells, you maybe transform them into an induced pluripotent state, or you put a transgene in that you can induce, and you can make these changes. You can also do things with defined medium, which we'll talk about today. So the HyenaDNA paper from the first week that talked about defined media and kind of getting the perfect formula for your medium and then using that. Then this is something that goes back to making induced pluripotent cells, where some people use transgenes, some people use a defined medium of a certain type. And I can tell you that there's a lot of sort of technical gamesmanship there. So, finding the right medium is great, but some people, there's a lot of skepticism surrounding some of the results for both transgenes and defined media. So, when you read these results, it's amazing stuff, but you can also ask the question, are we really getting the thing we think we are? We're getting, differentiated cells, we're getting tissues, we're getting things like that. But is this really translatable to, say, like a whole embryo setting, where you'd actually be able to make some primate, or maybe clone a primate, or create a primate embryo that could be implanted, which we talked about in the first week as well. So with that in mind, let's go to the papers. So this is actually an article in, I think it's in the Observer. It's a UK publication. This is by Philip Ball. This is a feature that they had on this human embryo models work. So this is kind of considering some of what's been going on. They showed an example of how these embryo models are made. So these are embryo models. They're not necessarily embryos. They're technically embryoids, or they're technically something similar to embryoids. They could be a gastroid or even just a standard organoid. Although what they're trying to do is build this embryo with the tissues and with the structure of an embryo, as opposed to just creating maybe like germ layers. But they are creating germ layers as well, as we talked about in the first and second weeks. So, they're creating these embryo models out of stem cells. They're transforming them to these different fates. They're building, the germ layers, and they're building differentiated tissues. They're building a structure that looks like an embryo. And so that's where we are. And there are a host of ethical challenges surrounding this. I don't want to discount those. This article, I guess, talked about these. Rare to see like a print version of an article anymore. And you can't really read the text, but basically goes over the papers we talked about in the first week and some of the details surrounding that. So if you can find that article online, that would be where that would be. So the first paper I want to talk about is this paper. This is a bunch of authors here. Islop et al. And the title was Modeling Human Post-Implantation Development by Extra Embryonic Niche Engineering. So this is the work on defined media. And the idea behind defined media is that you have the cells grow in this medium, and you have to find the right recipe. So it's a chemical inducement of this to create these embryo models. You have to find the right chemical formula. And so there are all sorts of growth factors in media. There are all sorts of chemicals that keep the pH at the right level and things like that. So there are all sorts of things that we can modify to optimize that environment. And that's what they're doing in this paper. They're trying to they're talking about this in terms of extra embryonic niche engineering. So extra embryonic just means out in the environment of the cell or of the embryo. And so when you're out in the environment of the embryo in a biological context, that might be implanting itself or being in the uterus or something like that. Now, the environment is, we talked about environment before in the group, and that is to say that it's hard to define. You have to really define what you mean by environment, which is, of course, a concern here because this is a chemicals formula for the environment, basically. So this is where this is one approach to making these. So the abstract reads, implantation of the human embryo commences a critical developmental stage that comprises profound morphogenetic alteration of embryonic and extraembryonic tissues, axis formation, and gastrulation events. So you're not only creating the tissues, you're creating the axial polarity. So things are towards the head or towards the tail in that sense. So they have to be in the right order. They have to be in the right place, not just differentiated. Then you have these gastrulation events. So like I talked about in the case of some of our topological networks, you have these shape transformations that need to happen, and they're very specific shape transformations. Our mechanistic knowledge of this window of human life remains limited due to restricted access to in vivo samples for both technical and ethical reasons. So we can't necessarily create like something in the womb that's frowned upon, but we can create it in a three-dimensional culture. This is why we were interested in embryoid models, because we can't experiment on fetuses basically, and there's a lot of this is actually why we have induced pluripotent cells, because there were a lot of ethical concerns and people have moral objections sometimes to stem cell models, where you're actually harvesting them from the embryo. And so that's where a lot of stem cells come from. They come from umbilical cord, they come from the embryo. And so this is a thing that's been, at the fore of this field for a long time. So this gives us a model where we can work on these problems without some of these ethical concerns. Additionally, human stem cell models of early post-implantation development with both embryonic and extra-embryonic tissue morphogenesis are lacking. So we don't really know anything, like I said in the first week, about post-implantation development. We know a lot about pre-implantation development in terms of like working on it in culture and in models, but this post-implantation world is quite different. Here we present iDiscoid. So it's a small I discoid, and it sounds like something Apple would make, but it's actually a biological thing. Here we present iDiscoid, produced from human-induced pluripotent stem cells via an engineered synthetic gene circuit. So they're actually using a synthetic gene circuit to sort of help us, help the embryoid model respond to its environment, the optimal environment conditions. So you have this, you're basically creating an induced pluripotent stem cell, you're using an engineered synthetic gene circuit, you're using that to sort of control the fate of the cell. iDiscoids exhibit reciprocal co-development of human embryonic tissue, an engineered extra-embryonic niche, remember all these chemicals, and a model of human post-implantation. It exhibit unanticipated self-organization and tissue boundary formation that recapitulates yolk sac-like tissue specification. So this is at a certain point in development where we get this yolk sac, we get the tissue specification, the boundaries of tissue start to form. And this is all sort of self-organized. There's no, there are chemical signals within the embryo, but critically there are chemical signals outside the embryo that are sort of enforcing this. So these yolk sac-like tissue specifications are extra-embryonic mesoderm and hemopoietic characteristics. Hematopoietic characteristics being the formation of blood. I think we talked about that in the first week. That was one of their hallmarks, is where they were actually developing, they were looking at how the blood, the circulatory system develops in the formation of blood. So the hematopoietic characteristics are the formation of bilaminar disc-like embryonic morphology, which is, your red and white blood cells. I think the red blood cells are what they focused on in the papers in the first week. The development of the amniotic-like cavity, and acquisition of an anterior-like hypoblast pole and posterior-like axis. iDiscoids offer an easy-to-use, high-throughput, reproducible, and scalable platform to probe multifaceted aspects of human early post-implantation development. So we can do a lot of things with this. We can do drug testing, developmental toxicology, and modeling diseases that occur in utero. So here are some images of what they're doing here. This is where they have 0% IGATA6 high supplementation. This is, okay, so this shows some of the features in here. Here's the hemoglobin starting to form here. This is another example of hemoglobin. This is day 12 of development. So we talked about this being the window when these embryoids are probably, going to give you the best window into development. So you have some hemoglobin signal here. You have some IGATA6 high supplementation, 25%. So this is 0%. This is 25%. This is showing some of these hemoglobin in context here. So here's some of the structures here and the hemoglobin surrounding it. Here's some more pictures of where you have kind of you're going through the structure from the mesoderm to the endothelial to the endoderm, so from the bottom to the top. And here's some images here where you have stain for FOXA2, CD34, desmin, which is a muscle thing, and F-actin, which is another muscle thing. So you see a desmin is in here being expressed. FOXA2 is in the blue. See some of this in the endothelial and especially in the endoderm. In the mesoderm, you have a lot of desmin, which decreases as you go towards the endoderm. And then CD34, which is another marker, which decreases as you go towards the endoderm or the top. So you get these three germ layers, these three types of precursors to the tissues, and you get different things expressed. So you get things expressed with respect to muscle and some other things that are in the, so you can see that they differ by where you are in the structure. Same thing here, you have desmin in white, you have CD43 in yellow, and then CD31 in red, and you see that as you move through from the mesoderm to endothelial hematopoietic cells, that it's changing its composition. So this is just an example of how these things form in time. This is actually an example here of what's happening in these embryoid models. This is a figure where they show the first symmetry breaking here, and then you see confinement and migration, so cell migration is happening. And then you have this bilaminar boundary. So this is corresponding to differential gene expression. So you have, so first of all, you start off with these two cell lines, the IGATA6, which are uninduced in the wild type. Then in the idiscoid cell line, you have the combination of them. So you're inducing the IGATA6, but it's also mixed with wild type cells. You have the symmetry breaking between the IGATA6 and the wild type. You have this confinement and migration of different IGATA6 cells. You have this bilaminar boundary between the GATA6 being expressed here, and in the wild type you get nanog expression, which is, these are, nanog is of course an important marker of differentiation and stemness. Luminogenesis is where you get more this sort of lumen forming in blue. This is another marker that they used for that. Amnion-like formation, you have the bilaminar disc, and you get another structure here of the amnion. So this is like the amniotic sac and the disc associated with it. The posterior formation here, then this is the model of the human yolk sac, and you're modeling hematopoiesis here from the mesoderm. So these are all things where you're starting to get structure, you're starting to get different structures you recognize as embryonic, especially human embryonic, and it leads to this model. So the second paper I want to talk about is self-patterning of human stem cells into post-implantation lineages. So this is from Nature, and this is an accelerated article preview. So they're going to have a different version of this out soon, where it's the final version. This is weird way they do this in this part of science where it's very competitive. They have these embargoed papers, they have accelerated papers. They try to bring them out all at the same time. It's just this is part of the culture. This article, let's go through the abstract. So the abstract reads, investigating human development is a significant scientific challenge due to the technical and ethical limitations of working with embryonic samples. So we've talked about how these embryoids provide an alternative to accessing experimental models that we couldn't access before. Here we show that human pluripotent stem cells can be triggered to self-organize into three-dimensional structures that recapitulate some key spatiotemporal events of the human post-implantation embryonic development. So this is where we're focusing on these spatiotemporal events, this organization in space, as you saw in the previous paper, and then over time. Importantly, our system reproducibility captures spontaneous differentiation and co-development of embryonic epiblast and extraembryonic hypoblast-like lineages. So we have two different lineages that are differentiating spontaneously and co-developing. This establishes key signaling hubs with secreted modulators, and then undergoes symmetry breaking like events. So symmetry breaking is where you have things that are very similar and they're identical, I guess, in terms of their spatial distribution. And then you get the symmetry breaking, which is where they differ. So like a left-right symmetry breaking would be where the left side differs from the right side. There's an event that triggers a difference. And these are common in development. This is involving, these signaling hubs where you get secretions of different chemicals out into the embryonic environment. So this isn't the medium, the extra-embryonic environment. This is within the embryo, the environment. And you get these secreted modulators that get pumped out into the local environment, and it tells other cells kind of what to do. So you want to establish these key signaling hubs. It helps to sort of organize the model. So self-organization maybe is a little fuzzy in this context, because self-organization just means that it's not involving some sort of supervised process. There's no top-down process. It's just kind of that these signaling hubs start to form. You get this secretome that sort of exists in the model, and then those secreted molecules actually help to, signal other cells and tell them where to go and what to do. So they use single cell transcriptomics to confirm differentiation in diverse cell states. So we were using a lot of labeling in the last paper, a lot of fluorescent labeling. In this case, they're using single cell transcriptomics. This confirms a lot of these sort of cell fates, that they're actually forming different types of cells. Now, how much of that type of cell it is something we don't know. So we don't know if it's exactly like the cells you see in the embryo or something that resembles them. So this is an important point, because sometimes these cells aren't identical to what we would find in an embryo. And as a result, we may or may not be able to use them for certain end goals, like drug discovery or whatever. So this is, these papers always have very detailed figures. And so this is just a sample of what they have to offer here in terms of their evidence. You have a couple things. First of all, I want to highlight the epiblast and hypoblast layers that form first, and then you end up with these more detailed structures. So here's an example here at Carnegie stage 4. You have this implantation stage where you have the inner cell mass. In CS5, you have this epiblast and hypoblast form around within a trophoblast, and then you have the amnion forming, and you have this more detailed structure. So you can see that there's this sort of layering and pattern formation that accompanies this protocol. Then they show some stains here. Go to the second figure where they actually do some gene expression. So they're doing this gene expression profiling, and they're plotting it out in a UMAP space here. So they're actually showing some of these cell types in a three components of a UMAP analysis. So they're showing that there are differences in the expression profiles, that there's some overlap, but largely it's, clustered in the space. So they'll call it clustering per se in UMAP. But this shows another analysis on two UMAP components where they show some of these cell morphologies. And this actually shows like kind of some of these differences where you have mesoderm here, you have extra embryonic mesoderm here, you have blood progenitors, as we saw in the last paper, trophoblasts, amnion and non-neuronal ectoderm, epiblasts, and hypoblasts. So these are all separated out in this UMAP space. So this is dimensionality reduction. You're basically trying to find how they, if they're separate or if they're sort of commingled with each other. And that's how you can tell generally how, different labeled cell types are. So they may identify the cell types from some other criterion, like staining for markers. And then they look at this gene expression profile, which includes, thousands of transcripts, and they can actually do this sort of analysis. So it's good that we have the next-gen sequencing and the dimensionality reduction here. And then this just shows more examples of some of the structures that are forming in the embryoid. You have these different stains for F-actin, DAPI, and AFP. So you can look at like how some of these things are expressed in the structure. And so this is an example here in G of where you get this sort of process here. So you get the HPSCs, you get the self-assembly stage, which is where, you know, they kind of come together in different ways. The self-sorting stage, so they're kind of forming an aggregate here from these individual cells. Then they're self-sorting into these layers. They're self-patterning where the layers start to pattern. And then you get these secretion centers here and here. And then these secreted inducers are pumping out secretome, and they're inducing other cells to form other things. So that's basically the process here. So you can see is you get this aggregation, you get patterning, then you get further patterning from sort of these hubs of secretome. And so this shows again what this looks like. You have epiblast-like cells, hypoblast-like cells. They get transformed through FGF and nodal action. It's in this sort of aggregation step here. And then you get the sorting of cells. So once you get an aggregation of these cells, you know, they're sort of a mosaic. They get sorted into these layers by gene expression profile or by how much FGF and nodal they're producing. And then they start to form this patterning, which leads to these centers forming. So the hypoblast-like cells either go to this anterior visceral endoderm-like cell fate or this amnion-like fate, and then it goes to a mesoderm-like fate if it's given enough BMP4. There's feedback of BMP4 for amnion-like cells, and then BMP2 from hypoblast to amnion-like cell. And so there, you know, BMP2 is playing a big role here in some of these transitions. One last thing to note about this is that they use the term like. So epiblast-like, amnion-like, mesoderm-like, those are all, I guess they're hedge terms, but they're also very descriptive in the sense that they're like those cells, but they aren't necessarily those cells. We're just assuming that they are based on their gene expression profiles and the way they behave. So you can see this human embryo model here, where you see this sort of diagram, this process diagram, translated into the topology of an early embryo. And then the final paper is this paper on gastroloids. This is in Stem Cell Reviews and Reports. This is on these gastroloid models. So I said that they're embryoids, and then they're gastroloids, which are a little bit more specific. So these are novel systems for disease modeling and drug testing. So I'm just going to go over the abstract here. This is a review paper, so I'll make this available to people in the group. Just kind of goes over the state of the art in this field of gastroloids. So by virtue of its inaccessible nature, mammalian implantation stage development, which is what we've been talking about, has remained one of the most enigmatic and hard to investigate periods of embryogenesis. Derived from pluripotent stem cells, gastroloids recapitulate key aspects of gastroless stage embryos and have emerged as a powerful in vitro tool to study the architectural features of early post-implantation embryos. So they're focusing on this stage with these gastroloids. And, you know, it's just really kind of recapitulating this process, which is actually very important to understand. And you can see that there's a lot of self-organization, and there are a lot of things going on. So it's important to have a very specific biological model that we can use for this. While the majority of the work in the emerging field is focused on the use of gastroloids to model embryogenesis, their tractable nature and suitability for high-throughput scaling has presented an unprecedented opportunity to investigate both developmental and environmental aberrations to the embryo as they occur in vitro. So we want to look at some of these aberrations in development, maybe like things, you know, dysregulation of some of the factors that we talked about, like BMP4 or something like that. When you turn it off, if there's a mutation that turns it off, or whether there's a circumstance that turns it down, what is the effect on the gastroloid or the embryoid? Or if the environment's different, if the environment is not as it should be. We talked about within the embryo environment, which is this secretion of molecules and signaling molecules that guide cells to their fate, or the external environment. So if the medium is not as it should be, and the medium is just a stand-in for the environment in the womb, because we can't necessarily know exactly what that's like, but we can try to approximate it. So we have these two different types of levels of environment, and if they have aberrations for some reason, then that can affect it. We want to know what that looks like, what those effects look like. This review summarizes the recent developments in the use of gastroids to model congenital abnormalities, their usage in teratogenetic testing, and the current limitations in this emerging field. So you can actually form teratomas and look at some of these congenital anomalies using these models. And this is, again, this is stuff that is important for medicine and important in other areas as well, like drug testing. So this is the review. I'm not going to go through the whole thing. I'm just going to say that basically this period of development has been kind of like a black box, and now we're kind of making this into a gray box, as they say in cybernetics, meaning that we know something about it now, but we don't know everything. And so that's work to come. So thank you for paying attention, and hope you learned something.